a combination of computational and experimental techniques. And I will leave the stage to Professor.
Thank you so much. Thank you all. It's really a pleasure to be here. Can you hear me well? I don't think this microphone is on.
Yeah, use that.
Oh, I guess it's not for you guys, it's for the people online. Is that the idea?
Yes.
All
Right.
Sounds good. So, yeah. Welcome. Who's excited to be here? Yay! Awesome. So I just came back from 40 student presentations at MIT that started at 1. We're supposed to finish at 2:30, but then went until 3:05. So here I am on my bike. Who's eco-friendly and faster? Yay! Oh, the mic's not on. Okay, awesome. Who missed all the jokes online? All right, so what I'll tell you about today is our own journey in transforming the way that we think about biology. And I'm going to put it mildly and provocatively, hopefully. The human language is a very simple construct. It is constrained by the fact that every human has to speak it. Yeah, even those people, you know. And it's also constrained by the fact that it has to be computable by our own brain. And our brain is constrained by evolution. There has to be some kind of linear progression of increasingly advantageous traits that eventually leads to human cognition. And therefore, the complexity of human language is constrained by that as well. And the last constraint, no, not the last, an additional constraint is that it has to run on 3 watts of power compared to our compute models out there. And then the last constraint, perhaps the most challenging one, is that it has to be redeveloped from scratch at every generation from one cell. We have to effectively rewire the entire epitome of human, I would say of evolutionary cognition from scratch at every generation from a single cell. And therefore, with only 20,000 genes, we have to encode not just the entire body plan of the human body, not just the entire immune system and the vasculature and the bone system, muscle system, but every single neuron and all paradigms of neural connections. So I challenge you to build a computer like that, and we have inherited one. And I think this is like one of the most extraordinary achievements of evolution, if you wish, that brought meaning to the cosmos. Again, to put it mildly here. So human language by itself is amazing, but what's even more amazing is that we're able to actually understand languages that we did not evolve, we did not invent. The human language is an evolutionary construct, and it's also an invention of an API for humans to communicate with each other. And therefore, by comparison, it is much simpler than a lot of the biological natural world out there. Now, let me also contrast the complexity of chemistry, of biology, of protein folding, of cellular networks, and of course, neuronal networks. Contrast that complexity with the complexity of physics. The rules of physics were written about 13.8 billion years ago. And last millisecond, maybe whoever was around at the time scribbled a few things and changed a few rules as the Big Bang exploded. And a few of the rules that were true in this extraordinarily dense early cosmos perhaps were slightly adjusted. But since then, God or whoever it is hasn't really changed the rules. There's just a small number of equations, and it's just running that model forward. By contrast, think about the rules of biology. Every single evolutionary niche that gets created establishes a new realm of physical laws, if you wish, upon which biology then responds. Biology is also not just 70,000 years old, like human language is, but it's like 3.4 billion years old, almost as old as our planet. So biology has been tinkering with functions since the dawn of time, at least the way, the time on Earth. And therefore, I want to pose to you that much more complex than ChatGPT, much more complex than understanding all of human thought, again, with all due respect to our own species, is the challenge of understanding biology, understanding chemistry, understanding evolution, understanding neuronal circuitry, understanding the biological world. And if mathematics is well suited to the rules of physics, that again were scribbled by Maxwell's predecessors many, many billions of years ago, those same rules that Einstein and Maxwell and Newton sort of, got progressively closer to, and Schrödinger, etc., those rules are well modeled by mathematics. But the rules of biology are well suited to a very different kind of thinking. And that kind of thinking should match the complexity of the tinkering of biology. And if you look at the tinker of biology, what is it? It is constantly adjusting billions of parameters to match some kind of functional landscape of selective fitness at any one time. Okay? And that's the challenge ahead of us. So AI has been amazing, don't get me wrong. ChatGPT is the most extraordinary invention that humans could have imagined. And even 3 months before it, I would ask the most forward-looking thinkers in AI, they had not seen it coming. None of them had seen it coming. And it's a major advance. Again, don't get me wrong. But perhaps the most lasting impact of AI on the human condition will be what we're doing right now in medicine, in biology, in chemistry. And I hope that you guys are as excited as I am about this. So with that, let me tell you about the work that we're doing in my own team, similar, of course, to what many, many other labs are doing, but a bit better, you'll see now. On using AI to understand, number one, health, and the medicine, the patient records, the patient trajectories. Number two, how to develop the next generation of therapeutics. And number three, how to understand the basic foundational circuitry of both human biology and of human disease. This talk can only go downhill from here. I've kind of promised it all. Thank you. This is it. All right. So the first part that I'm going to be talking about is the whole path from genetics to genomics, to epigenomics, to transcriptomics, to understanding the circuitry of the cell, understanding targets that we can modulate in that circuitry based on the wiring of every cell in the human body, and based on the genetic association of every one of 140,000 loci associated currently with human disease and human phenotypes, to then be able to understand where to intervene, and not just where to intervene, but how to intervene. And that's where AI for drug development, AI for designing the next generation of drugs comes in. But again, we don't want to just design it, we also want to put it into cells, put it into animals, and eventually put it into patients. The second part is understanding multimodal trajectories of humans. Being able to take this language understanding, this extraordinary advance of large language models and transformers, and couple them with structured data, multimodal representations of patient state to understand together how language and medical information are in fact painting a much richer picture of human patients. And lastly, about foundational AI, using not just tailored models to any one problem, but using foundation models with these extraordinary emerging capabilities that truly understand, that have the ability to give us new insights on drug discovery, protein function, and patient states, namely spanning the whole gamut from the structure of chemicals to the language of genomes to the structure of proteins and ultimately the phenotype. So as an ancient Greek, a descendant, I can't help but start with my ancient Greek ancestors. So here's a representation from Epidavros, right around the corner from Athens, where presumably a god was sort of observing some kind of patient, and you have these priestesses and this peer review committee on the side observing, and you can see that really not much has changed, except that instead of gods we have, well, physicians that are treated like gods sometimes. So this infallible view of medicine was completely abolished, of course, with the Renaissance. And this whole concept that disease doesn't happen at the human level, but it happens at the cell level, and the ability to look inside cells. And that basically takes us to a disease that is perhaps one of the greatest plagues of our society today, Alzheimer's. And Alois Alzheimer, who basically was able to diagnose nearly everything, that we define Alzheimer's with today just by looking into the microscope. So the importance of data, being able to look at the actual mechanistic basis of a disease, and then recognize the amyloid beta plaques and the neurofibrillary tangles that still define Alzheimer's today. And boy, have we come a long way. On the bottom right, I'm showing you 4 cells and 2 phenotypes, A beta and plaques. On the bottom right, I'm showing you 2.4 million cells, several orders of magnitude, about 6 orders of magnitude, and 430 donors. And every dot is not 4 phenotypes or 2 phenotypes, it is 20,000 phenotypes. And these are the expression values of every single gene. So maybe an extraordinarily smart human could understand the stuff on the bottom left and come up with Alzheimer's disease as a new diagnosis 100 plus years ago. But today, I don't think there's a human in the planet who can understand this amount of data. And that's one reason why AI is important, because the scale of the data is beyond comprehension. But as I argued earlier, perhaps our brains are well suited for human language. But there's really no reason why we should have any intuition about protein structure, about chemical structure, about patient states, and so on and so forth. So that's where AI comes in. Number one, the sheer scale of the data. Number two, the new deep integration. And number three, the representations that can bring multiple lines of evidence across things that we frankly have no intuition. And now in the middle comes a new, very, very powerful thing, which is beyond correlation. So both on the bottom left and on the bottom right, we have correlation. We basically have that, there's amyloid with Alzheimer's. It doesn't say that amyloid causes Alzheimer's, it says it's amyloid with Alzheimer's. And, there's been a big debate as to whether people died of COVID or with COVID. And whether, there's another study yesterday that basically says that it is grossly dangerous not to drink overly too much coffee every day. so I don't know if you saw the study, but it basically says that 3 cups of coffee dramatically reduces every aspect of, aging, cardiovascular, and you name it. It sounds great. It sounds great. But the question is it that people who drink a lot of coffee have like really high pressure jobs and they're working all the time, they're super driven, they have amazing healthcare, and you name it? Or is it that really coffee conveys these benefits? And if you go to Starbucks, 7 times a week, I assure you, you will have better health outcomes. But the people who will not take my advice are the ones who can't afford it. And they also have, no gym membership, and they also have, no healthy food access, etc. Okay? So the problem with both bottom right and bottom left of this picture is that it is about correlation. It's things that change along with the disease. But the middle bottom panel, that's a whole other world. This is the world of causation. It doesn't matter how weak the effect of a genetic variant might be. It gives us causality. So many drugs have failed because if you use a mouse model to show that you can reverse any process like cancer or immune or neurodegeneration in a mouse, it's not clear that that effect will also be there in the human homeostatic system. In other words, we have all kinds of feedback loops. You go and change my cholesterol, I bet you my gene expression will change to, put it back where it was. I go on a diet, no worry, my metabolism will change to put me back on the weight that I had before. The human system is extraordinarily homeostatic. No matter how you push it away from homeostasis, it goes back. And that's where genetics comes in. That's where the ability to say that no matter how small, this one genetic variant has an effect that penetrates straight through all of the layers of feedback and control that humans have to then take us all the way to a phenotypic consequence. Is everybody with me? And why do I care about genome-wide association studies? You might tell me, 0.1 millimeters in height. Why do I care? Because that's what evolution tolerated. Evolution tolerates tiny little changes. If I change things more, then that genetic variant will not rise to high frequency. So almost by definition, common variants have to be weak effect. And the strong effect variants will not rise to high frequency. Okay? So we have an enormous opportunity here in the middle panel to basically use genetics to understand causality, but an enormous challenge associated with that, which is that after we find the genetic locus, we still need to understand where and how to intervene at that locus. Why? Because 93% of the genetic associations with human disease, 93% are in fact outside protein coding genes. We have 20,000 protein coding genes. They cover 1.5% of the human genome, but only 7% of the disease variants lie within them. The other 93% do not perturb a protein coding gene directly. What does that mean? That means that I heard something about percentages. Yes, it's an enrichment. Only 1.5% accounts for 7%. So that's an enrichment. But there's still the other 93% to go. So if you care about the mechanism underlying human disease, you gotta figure out how to connect the bottom center to the bottom right. Okay? And that's basically what we're doing. Okay? Who's excited so far? Yes? Okay, so what are the shifts that are happening? How do we put all of these things together? Okay? 3 shifts. First, we're going from hypothesis-driven research to data-driven research. Okay? I like to call it, you shoot first, you ask questions later. Okay? Gather a ton of data, and then it doesn't matter what you need to know. You ask questions over and over and over again. And that's what we've done. We basically gathered massive amounts of data, and now we're writing paper after paper after paper, reanalyzing these data, because frankly, we've gone ahead and gathered everything I was hoping, we had. And that, of course, comes with systematic data set, that comes with building infrastructure, that comes with massive data sharing, that comes with comprehensive use, and, all of these things that, I think Danaher knows so well. Okay? The second shift is what I mentioned earlier. So I'm trying to sort of reduce the size of all of these. There you go. The second is we're going from correlation to causation. Okay? So genetic variants and perturbation experiments is something that we uniquely have in biology. If you find in most of human epidemiology correlations, there's not much you can do about it. But when you have this ability to go in and molecularly intervene, I think that that's a big deal for the perturbation experiment, and that's also a big deal for the genetic variants, its ability to have causality. But again, as I mentioned earlier, both the blessing and the curse of genetics is that it's mechanism agnostic. In other words, when you first find it, so powerful, you don't need to know how it works, and yet you find the association. But the trouble is, after you find the association, you still have no idea how it works. You now got to figure out and translate that to mechanism. And then the third dramatic paradigm shift, and of course I'll tell you about how we deal with number 2 as well. So the third paradigm shift is going from classical data analysis, where you basically hire a super smart statistician for every new problem you have, and they tell you, oh, well, correlate that with that and make sure you don't have too many variables, to generative AI, where 5 billion parameters, no problemo, give me 10. So why is this possible? Why is generative AI, deep learning, and these foundation models so ignoring of the number of parameters? The reason is that you are basically building generalization from the get-go. In other words, you're training them to understand the world. You're not training them for a given task. And that's why we call them foundation models, because nearly every one of their capabilities is an emergent phenomenon. The same way that us understanding mathematics or physics or how to use computers, it's an emergent phenomenon. We did not evolve for that stuff. It was not around when we evolved. We evolved for social behavior, for language, for dancing, for mating, for protection, for chasing, for scheming, for lying, for dissuading people and persuading people. That's what we evolved for. And yet, the natural best solution that evolution came up with to solve this set of tasks was a generalizable model that can now do all kinds of stuff that frankly, we were not meant for. Everybody here? So in that context, foundation models are very much like us. They're self-supervised. What do I mean by self-supervised? Who has had kids here? Awesome. Have you noticed the first few months of life, they're just like staring at the ceiling? What are they doing? They're training their edge detectors. So we have a general neural network that builds primitives and builds on those primitives to then do the amazing tasks. Who has heard of the concept of neoteny? Neoteny, holding the youth, holding the young. Greek term. Neo means young, and teno means to hold, like the tenets. So neoteny simply means that we, super advanced primates, and most primates, hold our young for a long time. A giraffe baby falls off, and literally falls off, and has to run, and it's fully developed, it's ready. Human babies, it takes about, I would say, 18 years for them to fend off by themselves. So what do they do during those 18 years? They basically are building higher and higher level of abstraction representations of the world. And how are they doing that? Self-supervised learning. Who has played peekaboo before? Peekaboo is self-supervised learning. Basically says that I have a model of the world for what the next action will be. And as soon as mommy reappears or daddy reappears, it's exciting. Because I don't have that continuity. The length of training of the number of parameters, the number of tokens, if you wish, in sort of whatever the hardware of a baby can hold, just not long enough. And therefore, the surprise is there. And how best to train this extraordinary neural network that we have inherited? It is by constantly making predictions about the future and updating those predictions as soon as the data becomes available. It is basically the world's best self-supervising machine. And what makes foundation models so foundational and so generalizable is that they work in very similar ways. How is ChatGPT trained? How is a large language model trained? How are transformers trained? You basically take a sentence of Shakespeare, for example, and you hide one word or 3 words at random. And then you ask, predict those words. Many people tell me, ChatGPT, what do I care? It just predicts the next word. No. That's the pretext task. We don't care about predicting the next word. What we care about is that if it gets so good at predicting the next word, in no matter what text, to get at, 60% accuracy, just look at word frequency. 65% accuracy, look at Kamers and, be a little cognizant of grammar. 70% accuracy, well, you better understand something about context, something about theme, something about tone. 80%, 90%, 100%. By then, you better understand the human psyche. You better understand the struggles that humans have, the, imaginations and the dreams and the fears and the theorems and Euclides and you name it. Is everybody with me here? So it is a pretext task. We are constantly teaching the model how to get better and better and better at capturing the world, all of the world. And it's the same thing with image models. You can hide a patch of the image and then say, what was there? Why is this a powerful learning paradigm? Because all I need to do is look around. I don't need any humans anymore to teach me what that label of this thing should be. I have basically self-supervised learning, where I can just let the machine loose and can like read all of our texts and then figure out how we think. See all of our images and then see what the natural world looks like. And then to get better at reconstructing that little patch of an image, well, if you understand about the continuity of color gradients, you'll do fine. But to truly figure out that there should be a missing window here, or that, between, I don't know, the top of this ball and the bottom of this thing that's hanging from it should be a rod, you need to understand physics and so on and so forth. Okay? And that's how these models are trained. Who's with me so far? Yeah? Who's excited? Yes? Have I scared you? Hopefully not. Hopefully excited you. So that's what generative AI and deep learning is about. It's about building these foundation models that are ridiculously generalizable, that are extraordinarily self-supervised, that are multimodal. What does that mean? So remember back in the days, we were talking about the fact that, you can look at a picture of an airplane, and the AI looks at this and says airplane. Then I can go in and mess up a few pixels that are imperceptible to the human eye, and the AI will now say giraffe. Okay, how is that possible? Because I can go and mess with the lowest layers of representation. And because I know how the system is working, I can figure out the activation gradients and then do exactly the reverse and then sort of figure out how to get a giraffe out of my pixels. So multimodal models that are trained on the combination of text and image are very robust to these kinds of attacks. And so are we. So we are extraordinarily robust because every time I say apple to my baby, it's looking at an apple, it's touching the apple, it's looking at the color of the apple, it's hearing the word apple, it sees the spelling of apple. And again, it's not something that our kids do naturally. They spend like a decade or more in school to even get to like conversing with us, at least intellectually interestingly. Okay? So this multimodal learning is extraordinarily powerful because if I have discrepant information, I can be much more robust to that discrepant information. Okay? And then the next key insight here is that it is about representation learning. It's about the fact that I'm not looking at the pixels directly. I'm looking at higher levels of abstraction, of edges and corners and objects and wheels and squares and eyes and noses and sails and so on and so forth to recognize any object. Is everybody with me here? So that ability that's why we call it deep learning, because it has multiple stages of abstraction. All of that leads to truly understanding the concepts. So we've gone from a world where the human scientist, however extraordinarily smart, does all the thinking, whereas now we actually have the opportunity to get new insights from these machines that are not constrained by all of the evolutionary constraints that we have, and the developmental constraints, and the language constraints, and so on. Everybody with me here? So I am so excited to be entering this new world where we are not the only thinking entity in this universe that we know of. I mean, this is amazing because we can learn new languages. We can learn multimodal representations of, say, protein structure and folding and language at the same time, of a knowledge graph with tens of thousands of genes and proteins and, drugs and diseases and anatomical structures, and throw in human language while we're at it. And that means that I can now speak with the machine and I can push the limits of human cognition further than they have ever been, than they have ever been. I think there should be an R somewhere. Anyway, yeah, further than, anyway. So are you guys excited here? Good. So you better be. If you're not excited, I'm saying it wrong. We talked about causality. We talked about this idea of I want to go here in the bottom center and figure out mechanism. Okay, so we have now this ability to find causal entry points to disease, but these causal entry points need to be translated. How do we do that? Well, we start, of course, with disease genetics because it gives us causality, and we include both common and rare variants, and then we profile molecular impacts of those genetic lesions. In other words, I know that this genetic thing here leads somehow to Alzheimer's, but I have no idea where and how it acts in the body. Or this thing here connects with obesity, but I have no idea what's in the middle. So we plug in the middle. We profile RNA We profile, oh gosh, you weren't seeing the same stuff that I'm seeing. So it's just the Zoom people who have a clean view. I apologize. Let me minimize some of those things. Sorry, I thought I was the only one bothered here. Actually, that's not even my screen. That's somebody else's screen. Oh well. Can somebody double click on that screen and make it bigger? I hope you guys can see. Yeah. Okay, so then you're not seeing my screen. I should stop that. Okay, great. So we need to understand circuitry, and we need to understand how that circuitry arises to understand the mechanism through which those genetic variants are falling. Thank you so much. This is much better. And to do that, we profile, and I'm sitting in a computational lab, but we have spent probably upward of $80 million over the last 20 years to figure out so many thousands of human patients at single cell resolution across their body, across their brain, across their immune system, and so on and so forth. So we have gone crazy in figuring out how to profile RNA, the epigenome, in both healthy and disease samples. Why do I care about healthy samples if I care about disease? Because I want to understand the impact of genetics. And if I have multiple healthy patients, healthy donors, some with the mutation and some without, I can understand what the impact of that mutation is in absence of disease, which is even more important than in presence of the disease. Everybody with me here? And of course, disease as well, because some of those changes will be impacted by the environment. So I can't just look at genetically driven changes. I have to look at both. So when I have all of this information together, I have the ability to now start integrating the data and then predicting driver genes, regions, cell types, regulators, tissues, and so on and so forth. Okay? And when I have that prediction, that's where it's time to take that other type of perturbation. The first perturbation is genetics, and the second perturbation is directed knockdown or knockout or overexpression or high throughput, screen, and so on and so forth. Okay? And of course, disseminate the results, start all over again. And we want to do that in cell cultures and in mouse model. And when I say circuitry, what do I mean? What I mean is I want to understand not just that there's something here leading to disease. I want to understand what are exactly the genetic variants. What are the regulatory control regions that those variants are sitting in? And in 93% of cases, it's not going to be protein coding genes. So it's going to be those regulatory regions. Who are the regulators upstream of these regions? What are the sequence patterns and motifs? What are the tissues and cell types where these enhancer and control regions become active? And what are the effector target genes through which these circuits are active? Everybody with me here? Awesome. So now let's, I keep talking about it. Is it real? So I was introduced as the author of the most depressing paper of that year when our paper in the New England Journal of Medicine came out about the mechanism underlying the strongest genetic association with obesity. Very often, still to this day, I go to talks and people say, well, there's 5 genes in this locus. We don't really know which of these 5 genes it is. In the FTO region, strongest genetic association with obesity, 1 standard deviation body weight for an adult in a homozygous setting. There was only 1 gene, and all of the other genes were far away. So people were so happy because they had it. They knew it was the FTO gene. And what we showed in that paper is that it's not the FTO gene at all. So here I am telling you that genetics is awesome and it's powerful and it works. And everybody was looking at FTO and I'm telling you it's not FTO. So what's going on here? What's going on is that, again, when it's a regulatory variant, as in 93% of cases, you don't know that the target gene is the one where that regulatory variant is sitting. Because sometimes that regulatory control region controls some other gene outside the gene that I'm sitting in. And that was the case here. The 2 target genes that we found, IRX3 and IRX5, were sitting 1.2 million nucleotides away, several genes away. And 600,000 nucleotides away. And the upstream regulator sits in a whole other chromosome. And we were able to narrow down the association from 50,000 letters down to 1 letter. And we were able to show that with that 1 letter, if I go and I use CRISPR-Cas9 genome editing to flip back and forth between the C and the T, then I can flip the cellular phenotype back and forth in a human setting. So that's why this is so freaking exciting. Because if I understand the circuits, I can intervene. And that's what we all want. We all want to figure out how is this very subtle change happening, and then can I go with a hammer and hit the same switch and then get a much stronger effect out there. Okay? And that's what this hammer does. So the question is, okay, this is the regulator, this is the motif, this is the enhancer, these are the target genes. But what's the mechanism? The mechanism in this particular case, we showed, is a new master regulator of thermogenesis, heat generation, the process through which our adipocytes are able to burn calories. By depolarizing the mitochondrial membrane, causing a loss of the proton gradient that is responsible for then creating ATP molecules and storing them into lipids, and then burning this very powerful source of energy as heat. Okay? So don't get me wrong, storing white adipocytes, I mean, that's wonderful. You eat Tuesday and you're still not hungry on Thursday. I mean, that's a big deal. It's a big deal when you don't have McDonald's all the time and when you're actually doing some walking. But with our lifestyle, a genetic variant that had zero association with obesity in 1930 became the strongest association with obesity in 1960. And how do we know this longitudinally? Because we can go back to the Framingham Heart Study, where we have blood samples from individuals who lived back in 1930, and we don't see any association with obesity. Because we have the phenotypes and we have the blood samples for these individuals. And we can do the association at every decade. And guess when this becomes an obesity variant? After World War II, when we're all sitting at the office and eating McDonald's. Okay? So phenotype is environment dependent. Beneficial or detrimental is environment dependent. In Africa, this variant is at 2% frequency. In Southeast Asia, 44% frequency. In Europe, 42% frequency. So it dramatically shot up in frequency, perhaps by drift, perhaps by selection. Going through deserts with very little food, going through ice ages. Maybe the ancestors that didn't have this fat storing mutation, or were less advantageous. Okay? And here we are today with the strongest association with obesity. And we have 7.5 hours, right? Until what time do we have? Over.
6 months.
Okay, so we now have the ability to now understand this. Who's having a good time? Yeah? Yay. We have now this ability to understand the circuits underlying these genetic variants. And we have 120,000 of these variants. And we have 120,000 of these circuits. We haven't gone and perturbed every one of them. Maybe that's what we should do together. Maybe that's what Danaher should be doing. Maybe we should just, go and solve it. We have all of the building blocks. So we can now go and intervene. That's why we're excited about circuits. Because if I just have this thing on the left, I don't know what to do. Yeah, maybe I'll knock down the FTO gene. Where's the FTO gene on the right diagram? Great question. It's not there. Right? So, but if I have the circuit, if I have the diagram, I know what cell type, what tissue, what regulator, what target gene, etc., 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 etc. Okay? That's why we're excited. So, editing a single nucleotide can now restore thermogenesis in individuals that didn't have it. What does that mean? That means that the oxygen consumption rates, both as basal as well as isoproterenol stimulated, can now be shot up by 7-fold. A single letter shoots up 7-fold. I mean, I don't know how much more precision medicine you get from one letter out of 3.2 billion letters in the human genome, knowing where to put the X, knowing where to, edit. I mean, that's a big deal. But that's the cellular phenotype. What about the organismal phenotype? Well, we took the downstream target gene RX3 that shoots up when this thing is busted, and therefore the regulator can no longer bind. It's not only a repressor. These genes get de-repressed. So what if we shut down these genes? In regular animals, what we see is that they become leaner to start with, using an IRX3 dominant negative construct under an adipose promoter. The animals start out leaner, the most impressive part is that we put them in a high-fat diet. Regular animals gain weight, as you would expect. These animals, they don't care. They just burn the calories. They're not exercising. They're not changing their nutrition. They're just burning more calories. If you look at the graph of diurnal calorie burned, they burn more calories in their sleep and when they're awake and all the time because they've basically flipped that switch and they're not able to store calories anymore. Who wants some of that? I know who you are because I'm homozygous double risk for this. My whole family's obese. I'm on the edge. So we could definitely use that. And GLP-1 is wonderful, but it's one mechanism. This is a whole other complementary mechanism that basically gets at the core of metabolism. Okay? So this is one example. Here's another example. ApoE4. 10-fold higher risk of Alzheimer's in a homozygous. I mean, this is dramatic. And it's high frequency too. It's crazy. So what does it do? The answer is almost everything. But at the molecular level, what we were able to show is that ApoE4, even in control individuals, leads to, in a cell type specific effect, an increase in cholesterol biosynthesis in oligodendrocytes. And this is true for Alzheimer's cases, but also non-Alzheimer's controls. So you see increased cholesterol biosynthesis in the carriers, but you also see less myelination in the carriers. So now cholesterol is one of the things that is being used by oligodendrocytes to coat axons with myelin, with this lipid-rich myelin sheath. So when you don't have myelin, the signals don't transfer as well, but also the neurons are much more vulnerable to damage. So you would expect to use cholesterol to make myelin. Then why is the cholesterol going up and myelin is going down? Remember the part about homeostasis? So you see that there's not enough myelin, let's make more cholesterol. Still not enough myelin, it's even more cholesterol. And what's happening to the cholesterol? It's basically getting stuck in the endoplasmic reticulum. ApoE is an apolipoprotein E. It's a lipid transporter. Guess what? Transport is broken. So then the oligos keep calling for more cholesterol, and the cholesterol is not getting to the right place. And that's by itself is a problem. But now if you now count all of the accumulation of lipid particles and all of the oxidative damage that you can have in the middle of these cells, this is compounding. So we basically said, okay, great. We think we understand a potential mechanism. What if we actually go and facilitate cholesterol transport with cyclodextrin? Boom, cholesterol transport was restored, myelination was restored, and in a mouse model, cognition was restored. The power of understanding mechanism. Okay? Immunotherapy, extraordinarily powerful. Metastatic melanoma used to be a death sentence. Now, for 50% of patients, you have remission. And that's amazing. We should be celebrating that. But for the other 50%, it's still a death sentence. So what do? We need to understand what's different between responders and non-responders. We can basically now go and study the epigenome of responders and non-responders to metastatic melanoma immunotherapy. And what we're finding is that there are hundreds of regulatory regions that are differentially active. There's a common set of regulators upstream of them. We took one of those regulators and we basically said, hey, let's flip these epigenomic regions for those patients that do not respond. And again, in a mouse model, we haven't treated any humans yet, of course, but in a mouse model, we found 100% success of immunotherapy in a combination treatment. Let's do that. So all I'm trying to say is that by understanding mechanism, we can go a long, long way. And I can go through more and more examples across a range of diseases, so obesity, Alzheimer's, cancer. But I want to step back and say, where do we go? What else can we do? I can sort of make you red in the eyes going example after example of that circuitry. That's a small component of it. Another giant component of it is that we think of Alzheimer's as one disease. We don't think of obesity as one disease. We think of cardiovascular as one disease. It's ridiculous. It's not one disease. It is dozens of different biological processes converging on a small number of common phenotypes. that the root causes are actually heterogeneous across individuals. So we can now use these same techniques to go after heterogeneity, to basically look at hundreds of patients and understand the difference between pathology and cognition, between cognitive decline and resilience, and so on and so forth. So we can basically find individuals where you have pathology, but you don't have cognitive decline, and try to understand what's happening there. And what we're finding is that there are very specific classes of inhibitory neurons that are associated with resilience. And specifically, these reelin inhibitory neurons are associated with resilience. I see you standing up. Do you want me to start wrapping up?
Yes, please.
Okay, sounds great. I asked earlier and I was told lots of times, so I did not speed up. So 10 minutes is okay?
Sure.
Okay, so 10 minutes.
Yeah.
Okay, so that's basically looking at reeling as one of these resilient pathways. And we can use this to understand systematically what are all of the hallmarks underlying disease. And we're finding mitochondrial function, synaptic signaling, cohesin expression, mRNA metabolism, lipid metabolism. And what we're starting to understand is that there's a small number of recurrent hallmarks of disease, and that these hallmarks are repeatedly reused in different combinations for every individual. So it's not that there's like 5 subtypes or 20 subtypes of Alzheimer's. It's that there's a small number of hallmarks, maybe 15, maybe 20. And one way to change dramatically the face of medicine is to stop thinking of diseases monolithically and to start going after these hallmarks. And maybe a challenge would be, what are the 3 hallmarks that we want to target this year and the 3 that we want to target next year, to start making a dent? And what are all of the diseases where these hallmarks are actually getting reused? Because lipid metabolism underlies Alzheimer's, of course, but it also underlies cardiovascular disease and metabolic disorder and obesity and all kinds of other systems. Now, one of these systems is also these cognitive resilience factors in astrocytes using choline metabolism as a new point of intervention. Where you can basically see here, everybody's talking about excitatory neurons, and that's the base of cognition. But also microglia, the immune cells of the brain. And we showed that like 10 years ago, that in fact the strongest genetic association with Alzheimer's across all of the loci lies in microglia, not neurons. And what we're finding here is that in astrocytes and oligodendrocytes is where a lot of this resilience sits. And we can actually start looking at this diversity of cell types. Now, microglia, they're only 10% or 5% of the cells. And within them, we can actually find that there are multiple states. There's 12 different states of microglia. And what we showed is that by switching the state of the cell, a different therapeutic modality altogether, it's not a drug anymore, it's a cellular change. And that cell state change was able to go from inflammatory microglia back to homeostatic, which basically was able to restore a lot of the, again, homeostasis inside the cells. In schizophrenia, we were basically able to show that these hundreds of genetic variants that have now been identified are localizing differentially expressed genes in specifically excitatory neuron subtypes in the brain of patients, in a paper that was just published in Science just a few months ago. In a paper that was just published in Cell a few months ago, we basically showed that ALS, a motor disorder, and frontotemporal dementia, a cognitive and personality disorder, are in fact showing the exact same mechanistic basis, and that both of them are affecting both regions, but somehow manifesting in only one of these regions. But it's the same long-range projecting neurons, which we call von Economou neurons in frontal cortex and dead cells in motor cortex, which are recurrently disrupted with the exact same orthologous processes. We're finding that in obesity, there are these metabolic hubs associated with this extraordinary adaptation that our species had to undergo to be able to power this organ that burns 20% of our calories. So we underwent massive metabolic adaptations, and these have happened in a small number of hubs that are now genetic centers of association with obesity. We're able to now study the process through which exercise is impacting your physiology, not just in your muscle and in your fat and in your metabolic organs, but even in your brain. We're finding changes in immune cells that are associated with exercise intervention and obesity interventions and diet interventions. And these are pointing to very specific pathways of hormone-mediated signaling, microtubule localization, giving us, again, entry points to understand what are the beneficial effects of exercise on the brain, but also across the body. I spoke earlier about these hallmarks of Alzheimer's. We're able to now take these hallmarks, 30 distinct hallmarks, and now start understanding, can we start predicting distinct phenotypes associated with hallmarks? And we have distinct combinations of genes associated with early or late, with female or male, with high educational attainment or low, and so forth. And this is actually one of the most powerful slides. We basically found that we should not be thinking of phenotype at the level of one person as a binary variable anymore. And we have a way to get out of that. Right now, in our cohorts, we classify Alzheimer's, non-Alzheimer's. But of course, people are a gradient, and somebody might be 80% Alzheimer's or 70% Alzheimer's. And moreover, a person who's 70% Alzheimer's doesn't have all of their cells at exactly 70% Alzheimer's. No, every person has a gradient of their cells as well. So what we have done is that by projecting the phenotypes of individual donors across 430 donors back to every cellular neighborhood, we're able to understand neighborhoods that are more Alzheimer's-like and neighborhoods that are more control-like. And the moment you've annotated the neighborhoods, it's a game changer, because you can now take all of the cells from a person, let's say patient 400, and that patient 400 has all of their microglial cells in red spots and all of their oligodendrocytes in blue spots. What does that mean? That means that perhaps the pathology of that patient is more associated with microglial dysregulation and less with oligodendrocyte dysregulation. Who's with me so far? So we can use that to change completely the paradigm with which we do single cell analysis. We can now look at drug response curves within a patient. We can basically say, what are the cells where I increase the expression of that gene or decrease the expression of that gene? And how does that correlate with all of the other phenotypes associated with that gene? And if I bring genetics and EQTLs and polygenic scores for gene expression, I can basically understand causality associated with that intervention. And I can basically get drug response curves within a patient in a human brain, something that no one has been able to do before. And we can also start studying disease progression within a given patient, because I can look at all of the cells of that patient and see what are the changes that are happening progressively, one after the other. And we've also used these language models to go and study the electronic health records and derive all kinds of insights. And we're also using the AI to carry out structural deep learning to understand the three-dimensional representation of molecules and of protein to be able to connect together these changes. And we're collaborating with glycogen in chemistry to actually synthesize a lot of these predicted drugs for intervening in specific domains of protein to basically start dramatically altering the circuits. And we're collaborating with Li Tai and Bob Langer to basically use these three-dimensional organoids that capture the neurovascular units, myelination, microglia, blood-brain barrier, astrocytes, and vasculature to be able to understand how we can actually model human-specific responses in a living organ. And this part is all about this pipeline, going from big data to circuitry to AI to organoids and to chemistry. I want to finish with just a little bit of an expansion in that bottom right corner that I was so excited about. I told you about this concept of representation learning, this ability to go from the pixels to the higher level of abstraction. We can do the same thing with DNA. We can go from DNA letters to words to grammars. In chemistry, we can go from single atoms to understanding every atom in the context of all the other atoms, and to understanding every amino acid in the context of all the other amino acids. And therefore start figuring out where in every protein is the function actually encoded. And that's extraordinarily powerful, because we can now start combining that with patient records and knowledge graphs and large language models and these cellular embeddings, and most importantly, carry out these multimodal embeddings that I was talking to you about earlier. And we have now used that to build a reasoning, interactive, user-guided, and AI-powered drug design platform that speaks the language of protein structure, that speaks the language of chemical structure, that speaks the language of single-cell biology and biological structures, and English, so that we can interact with it as well. And we have used this to start predicting new drugs in cardiovascular disease, metastatic melanoma, and Alzheimer's. And we're able to now not only let the AI combine all these different data modalities, but actually let the human back in. And that means that I can build this knowledge graph, and through this graph neural network, convolutional layers of abstraction, understand the latent representation, this Z vector representation of every gene. And projecting that very high dimensional representation to 2 dimensions basically shows you different gene functions. And those gene functions are associated with different structures. But I can't just do that with, I can not only do that with genes, I can also do that for diseases and drugs and biological processes and understand that language of biology that I started my presentation with. And we've built an interactive platform that is able to now let the human back in and let the human browse these embeddings. Here's an embedding representation of 200,000 drugs. And I'm using a variation autoencoder on a graph neural network representing the geometric constraints of the structure of every one of these drugs. And I'm able to now go and look at different parts of the chemical landscape and start asking which ones are associated with Alzheimer's function or lipid function, and so on and so forth. Which of these drugs are being used by your competitors or by your partners to study cardiovascular or Alzheimer's or obesity? And how can we now have a better understanding of that functional landscape? So this is, again, bringing the human back in, not just in a language chat kind of way, but in a visual way that basically allows humans to now browse. This is 3 million articles from the New York Times, where I'm able to go in and create ontologies. If I ask you to represent every single article that the New York Times has ever written, I mean, you'd be hard-pressed to do it. It took the system less than 30 seconds to do that, by basically going in and studying the latent representation embedding of how the AI understand that, and then bring the human in the loop. And we've done this for everything. We've basically taken every scientific paper that was written by MIT, every patent and every grant, and we've now created on the top left multiple floors with which we can now understand this information. And at the top floor, you have papers. In the middle floor, you have grants. And at the bottom floor, while we're at it, we took in the geographic location at MIT where these papers were actually written. So you can now start understanding how all of these relate to each other. In the bottom left, I'm basically showing you every class at MIT and the prerequisite relationships between them. Here I'm showing you how knowledge changes over time. And on the top right, I'm showing you quantitatively 100,000 loans clustered by their language and mapping quantitative information about them. And we've created a platform that I can't wait for you guys to use to study drugs, to study diseases, to study genes, to study anything, programs within the company. And we have this cognitive map. We have a lasso tool that allows you to select points. We have the ability to search and see the results visually within the search. We have this narrator, this AI model that is able to chat with you as you're focusing on different aspects and ask questions specifically about those. You can ask it, hey, how do you do this? And it goes and writes the code for you that you can edit on the fly. It basically is able to plot, and you can collaborate by bringing multiple people here. And you can now study these neighborhoods and launch agents and bring them back with an orchestrator. So this is, I think, the future of AI, where humans and AI are sitting side by side, where AI is not just sitting behind this extraordinary gate, and you can slip it little notes, and it can slip you back little notes with the answer, and it has access to all the data. No, we want to let humans back in the loop to study these extraordinary power that we have for understanding topology, understanding these landscapes. When you go skiing, every single time you look at the map and you understand the land masses, you understand the streets, you're able to abstract, simplify the landscape, summarize, make decisions. And that's what we want to do with cognitive cartography. So that's my vision for the road ahead, how we can take now this extraordinary wealth of data sets and these extraordinary machines that can be our partners to together study something well beyond the capabilities of our own minds, but not to replace human minds, but to augment them with powers that we never had before. And I think that we can enable personalized medicine, we can change the way that pharmaceutical development is being made, and we can basically bring together this coalition of AI, biology, chemistry, biotech, finance, pharma, patients, hospital, and basically change the way that we understand genetics and these hallmarks and patient subtypes and cellular biomarkers and these foundation models feeding into the design of the drugs and the interpretation of the organoid model and these cell-projected phenotypes to understand these drug response curves and so forth. So that's where I'll stop and thank this extraordinary group of people who are making this possible. Thank you for your attention and thank you guys for letting me run way, way over time. But hopefully there's enough excitement on the room. Who is very excited about what they see? Yeah. So much stuff every single year.
Absolutely fascinating, fantastic. Maybe one or two questions, if there are any questions?
Hi, excellent talk. Quick question. Excellent understanding, a lot of work, a lot of publication. I'm just curious, have you used these data sets or understanding to run a clinical trial to see how the patients are responding to these findings? And if so, what is that?
That's the challenge with academia. It's harder to raise $100 million. So, basically, we have done all of the preclinical work, and we are fired up and ready to go. So, I would love to work with you guys and take it to the next level. I would love to also take whatever partners you're working with, and I know that Danaher has this extraordinary reach about building resources that you can use to enable any one of your pharma partners, etc. And what we can do is basically go and advise on the probability of success of different trials by understanding the biology, or using the results to feed back and now start interpreting why did all of these drugs fail. I mean, the results of a clinical trial can be extraordinarily powerful in understanding these modulations and then feeding them back using this framework into these foundation models, these graphs, these networks, these representations. And I think that that's absolutely the next frontier. But that's a place where academia ends and academia industry partnerships begin. And there's no person better than Cheryl to tell you about how that's possible.
Questions?
Some really amazing work. It's really like almost have tears in my eyes. I'm actually,
I want the tears, I want the
yeah,
range of emotion.
I'm really curious about the computing power consumption.
Beautiful question.
You have done on this and also the scale of the tokens in this,
Yeah.
and as well as like how heavy is the, I would say, the human intervention of the annotation as well as the characterization, because I would assume it's not unsupervised, probably it's more supervised model creation. So it's like just want to get a feeling of like how rich and how crazy it is.
These are amazing questions. It is pretty crazy, but if you ask me how much compute we need, I only have one word: more. And it doesn't matter how much we have, and that's the problem. We always need more and more and more. However, the models are getting cheaper to train. There are plenty of pre-trained models, and we're doing fine-tuning. We're doing late-stage fusion. It's not as powerful as early-stage fusion, but it's still powerful because it allows you to unite spaces by building on the billions of dollars of training that have been collectively spent across our community. That said, we could absolutely use more compute, and we're basically training like 4 billion tokens right now when it comes to understanding protein structure and language together. We have a paper that's coming out on tomorrow, not today. We're posting this paper tomorrow with Marika Zitnik. That basically is now training this model that has both language and structure together, and it has its conversational abilities, and that allows us to pull at some. But what I showed you is a tiny little glimpse of what we want to do, and the model we're releasing tomorrow is just protein and language. But we also have these other models for language, for language and knowledge graphs together, and I think that the context of these models can absolutely increase. The number of parameters from those however many billion we have now can absolutely increase. I think that in the world of human language, we've kind of hit the limit of where we can continue seeing improvements. But in this multimodal world of structure and language and knowledge graphs and chemistry and biology and all of that, I think there's still plenty more to learn. So in terms of so I would say that this is where a lot of the strength is now. So the amino acid mutations, understanding the structure, understanding these networks and these modules, being able to sort of feed all of that through GNN, through LLMs, And of course, you asked, how much do you babysit these models? How much do you fine tune them? And that's where these platforms that I showed you for actually navigating these embedding landscapes come in. So basically, this Mantis platform that I kind of flashed at you, that platform I think is not necessarily for fine tuning, but it is for poking and debugging and opening up the hood and not having a black box model anymore. So basically, this turns your black box models into white box models, glass box models, because you're able to now ask, what have the embeddings really learned? Can I now start, asking about different neighborhoods in this embedding landscape and whether they truly capture what I expect as a human? And in some cases, we'll catch mistakes of the models. In other cases, we'll catch mistakes of our understanding of biology. And I think we're kind of open to both. But this ability to do this at scale, I think is extraordinarily powerful.
Thank you so much once again.
Awesome. Thank you. Thank you. Such a pleasure. Thank you for having me. Sorry for arriving just in time. Bye, everyone online.